So I'm a comparative animal biologist, and Joe Handelsman asked me to come here and take you back in evolutionary history and talk about where we came from. So the reason why this is important is because the human microbiome, we've only known really about this for about 15 years. And it's phenomenal that we've known about pathogenesis for thousands of years. We've known about the other organ systems for many, many, many years, hundreds of years, many of them. And yet, we're right. This is a complete revolution in my mind in thinking about animals and humans. I think humans have made two mistakes, in my opinion. One is to forget that they're animals, and the second is to forget that they're embedded in the environment and that they evolved as components of the environment. And I think these are important things to remember. And so what I'm going to do is, just for context for you guys, why don't you all sit back, and I'm going to give you a little biology lesson, take you back to introductory biology. And so we're going to be talking about the origin and evolution of the microbiome. And so what I'd like to do is I'd like to talk about humans as components of the animal kingdom. Who are we and how did we get here? And basically what we're talking about is the evolution of the epithelium, mucosal immune system, and bacterial, that bacterial consortium triad here. And this is basically the unit of selection on this system. So let's go back to the Cambrian explosion. So 3/4 of the way through the evolution of the Earth, all animal body plans evolved. And so this was about 540 to 520 million years ago. And the reason why I bring this up is because I want to emphasize that the vertebrates, of which we are a part, are not a crown group. We are actually, the vertebrates are down here at the very base. The vertebrates diverged around the same time that all of the other body plans diverged. So they didn't invent something new. They didn't, they don't have a better strategy. They just have a different strategy. Okay, so this happened, way back like 540 million years ago. And the other thing I want to emphasize is that over 96% of the animal diversity is non-vertebrate. In other words, every life history strategy known to man. And so when we start thinking about how we evolved to handle microorganisms, we ought to think about the fact that we just, like I said, have an alternative strategy. And all of these other organisms have their own complex evolutionary paths and, all that other sort of thing. So what I'm going to do now is some of you guys heard me present this yesterday, but I'm going to take you up this trajectory of human evolution at the genomic level, at the bioinformatic level. So you start here, and you're the metazoa, then the bilateria, then the deuterostomes, and the chordates, and you're up in the vertebrates and up in humans. And so there's a very, very interesting guy at Max Planck Institute. He's the head of the Max Planck Institute for Evolutionary Biology in Cologne, Germany. And what this guy does is he takes all of the sequence data available, and he has developed a technique he calls phylostratigraphy. And so one of the very cool things he's done is to ask the question of where the human genes came from, okay? So what he did was he took all the sequence data known, and what they did was they looked to see where all of the 23,700 human genes came from, okay? And what they were able to do is they were able to show that these genes showed up. There was a series of inventions 19 times. So you have these 19 steps. So it just a phenomenal effort. I think I was talking to him, and he said it took up all of the computer for a very long time. But the important thing is the origin of the genes so most of the genes, when you look at them by gene number, most of them are actually prokaryotic. So are we surprised, with oxidative phosphorylation and all the biochemistry that was invented with the prokaryotes? Most human genes date back to the prokaryotes. Then there was a big increase in diversity of the genes at the animals, at the jawed vertebrates, at the mammals, and then the primates. And so this is where you see the inventions in the genes, or the evolution of the genes that gave rise to the human being. Now, you could do this for any animal, but this guy did it for humans. Then he did a really interesting analysis. He looked at the 1,760 genes in a human, for which we know that if you make a lesion in that disease, you make a lesion in that gene, you get a disease, a genetic disease like cystic fibrosis or something like that. And there are 1,760 of the 23,000 genes that are known to cause a genetic disease. So he asked the question, when were those genes invented over evolutionary history? Are they, since these are human genes, are they human? Since these are human diseases, are they human genes? Well, in fact, what he found was that the genes that are associated with human diseases, and these various ways that he did, these are just ways that he analyzed the data, and they all showed basically the same thing. And what he showed was that all of the genes, almost all of the genes that are associated with human disease are ancient genes. Okay, so the genes involved in genetic disease are biased toward the ancient genes. And in fact, only 0.6% of them, in other words, 9 of the 1,700 genes are mammalian genes. All the rest of them are ancient genes. So the question comes, can we derive anything interesting about the human microbiome from looking at these patterns that Domazat, Lotso, and Tautz had derived? Well, if you look at this carefully, what I did was I went in and I looked to see which portion of these genes, I mean, at these inventions, are these genes that are, what are they involved with? And it turns out that when the animals evolved, you get a huge amount of signaling going on, and that certainly signaling with the microbiome would be important. Up here at the jawed vertebrates, you have an overexpression of the immune-related genes at both the jawed vertebrates and the mammals. So it looks as though many of the genes in the inventions that led to humans were driven almost very likely by interaction with the microbiome. So what this means then is that bioinformatic approaches to the evolution of genes suggests something in my mind that's very interesting. And that is that, given that over 90% of the disease genes have emerged before the bilateral radiation, it seems justified to use organisms that are evolutionary very remote. In other words, we can learn a lot by looking at how other organisms have evolved with their microbiome to provide insight into how humans have evolved with their microbiome. So I want to talk about, since this is about the impact of the environment on the microbiome, I want to talk about the environmental context. And this is to say, to the question of where did we come from? And what were the evolutionary selection pressures on what drove the mucosa, the evolution of the mucosa? So you go back, to this day, 96% of all animals are marine. And all of the 37 animal body plans arose in the ocean, okay, 540 to 520 million years ago. What were they doing at the time? Well, this is what they faced. 99% of the organic material in the ocean is in the form of dissolved organic particulate material. And it's really interesting. We have such a terrestrial bias that for the longest time, we didn't realize that a very large majority of the marine animals actually just take food across their body wall. They take up these dissolved organics and use those carbon sources as it's very rich environment. So in addition to that, they're sitting in 5 times, or 10 to the 5th to 10 to the 6th bacteria per mil of seawater. So they've got this huge bacterial pressure on them. So they're doing 2 things. So the evolution of animal body plans, there was a selection for the uptake across the body wall, and at the same time, they had to resist, they're competing with and resisting colonization by the surrounding microbiota. So, I mean, this is a tremendous sort of problem that they have as they evolve. So they're sharing the nutrient pool, and at the same time, they're trying to dissuade these organisms from growing all over them. So there are a series of very relevant milestones in the evolution of animals that I think are important when we think about where we came from and how what we're doing. So what I've done here is I'm indicating the major milestones in the evolution of animals. And the really fun thing is you go back to the sponges, and people like Uta Henschel in Germany are showing that sponges can be 60% microorganisms inside their cells and outside their cells. They can be, by weight, 60% microorganisms. Now, sponges are at the cellular level of organization. They have no tissues. Once you get up to the tissue layer, what you've done is you're a diploblastic organism. You have two layers, an outside layer and an inside layer. All of a sudden, you've got tissue layers, and at this point, you start not having a huge number of microbes associated with you. Then you become bilateral and triploblastic, and you get an internal layer that has no association with microbes. You get a complete gut, and then a coelom. And a coelom, what these do is you invent epithelia, you have internal organ layers, you have one way, I mean, imagine you, with the complete gut, you have regionalization. And that makes a huge change. And then finally, the evolution of the coelom accomplished separating the gut from the body wall so the gut could fold and become long. And so you have all of these. And when you think about each and every one of these inventions, they certainly, a significant portion of the evolutionary selection pressure was likely coming from interaction with the microbes in the gut on the epithelium and so on. So just to give you one example, Tom Bosch in Kiel, Germany is working on Hydra. Now, Hydra is one of these diploblastic organisms, and he's showing that many of the responses of animals to the microbial world are very ancient and conserved. So what he does is he likens Hydra to the human intestine. So the entire body of a Hydra is just like a human intestine. And it has two cell layers. Like I said, it has nothing but epithelium. What a phenomenal model. Nothing but epithelium. So it has an epidermis and a gastrodermis. And what Tom has done is he's done something that is, for the human microbiome, is going to be a long time in coming. And somebody asked this question. And with an animal model like this, you could ask this question. So he analyzed the phylotypes. And in invertebrates, it turns out the phylotypes there aren't a whole lot of them. So he looked at a series of Hydra species, and he found in each one 3 or so phylotypes reproducibly individual to individual. Remember, they're in a background of 10 to the 6 bacteria per mil. So and this was reproducible. Now, these animals had been in his lab for 30 years, and they'd been interacting with one another. And he would take a Hydra oligactus out, and it would have the same 3 phylotypes on it, reproducibly, and vulgaris, the same phylotypes. So what Tom did was he went out and he found in nature those very animals, and he showed that they had the exact same phylotypes that they had in the lab. So fidelity generation to generation was what he showed, which was a phenomenal thing. So there appears to be coevolution, and as Lita said, it's really important that we pick up these bacteria from our environment. But one of the questions is, to what extent are they co-evolved? To what extent is there fidelity generation to generation? And this is the kind of thing that points to the fact that animals were able to do this. Very primitive animals are able to do this. Also, Tom showed by doing a knockout in one of the response elements that's known as a response element to microbes throughout the animal kingdom, he has shown that from Hydra to man, the types of responses are conserved. And so it's just, these are really, really, really ancient things going on. So when you make the water to land transition, which is what we've done, the radiation of 7 of the 36 phyla, some of members of each one of these major radiations had groups that moved onto the land. I just want to remind you that said, in the vertebrates, more than 50% of the vertebrate diversity is in aquatic animals. So vertebrates are principally aquatic. In the water to land transition, land animals as plants as well required 4 things. They had to do 4 things. They had to solve four problems. One was they needed structural support to defy gravity. They needed to respire in air. They needed mechanisms by which to reproduce in dry environments, and then they had to avoid desiccation. So what this meant, of course, was, I call it inside out. What it required was a substantial remodeling of the mucosa, whereas many marine organisms will have their mucosa interacting directly with the environment. That does not happen once you've undergone the water to land transition. So we have all of our mucosa protected. That's not to say that it doesn't interact with the environment. So like I said, what we're talking about here is this triumvirate, but I think that our concept of the mucosal immune system is going to be the place that changes the most in the way we think about how animals work. And I think, as I mentioned, I think this is a revolution in biology. We've always thought of the vertebrate immune system as being a non-self-recognition system. And I think with the finding in the microbiome, we might want to rethink that. So I just wanted to take you back right here to the Cambrian explosion and to show you once again that the vertebrates diverged way long time ago. The presence of an adaptive immune system is not a new invention. It is not a superior invention. It is just an alternative strategy to interacting with the microbial world. So the adaptive immune system came in as the RAG1, RAG2 genes, likely bilateral gene transfer, came into the vertebrate genome and allowed for VDJ recombination and for memory in the immune system. So what you have here is you have all these groups, 96% of all the animals, not having an adaptive immune system, and the vertebrates, it falls in right here. And so that a shared derived character of all of the classes of vertebrates is to have an adaptive immune system. So what this shows when you look across the animal kingdom is that there are basic differences in immunity between the invertebrates and the vertebrates that, in my opinion, may reflect how they interact with microbes. A couple of years ago, I wrote a Nature concept piece in which I put this forward, and it was, I got a lot of tomatoes. and the fact of the matter is, though, I still stand by this, because as I look across the animal kingdom, this is what I see as I study the relationship of microorganisms to animals. And so what you see in the invertebrates is that binary associations, intracellular, extracellular, are very common. I've stood up in front of physician audiences and I've said to me, name me one vertebrate that has a beneficial intracellular bacterial symbiont. And I've rarely gotten one. Every single phylum of invertebrate that's been looked at has intracellular symbionts that are beneficial. And so there's something very different about the way vertebrates respond, and perhaps the way the adaptive immune system is shaped the way vertebrates respond. The invertebrates seem to have very rarely have consortia. Of course, termites are the exception. But very common in the vertebrates, a shared derived character in the vertebrates, is the carriage of consortia, very complex consortia. So the correlation in the immune system is that the invertebrates have the innate immune system only, and I would argue that they're stronger. I would argue that they just say no most of the time. They just say no to microbes most of the time. Whereas the vertebrates have an innate and adaptive immune system, and the price they pay, what they're gaining is probably some sort of increase in digestive efficiency and so on and so forth, and using microorganisms for what they're good for. But what they pay, how they pay, is that they're a little more permissive. And that permissiveness and the adaptive immune system causes autoimmune disease and so on. So I would argue that the principal selection pressure on the evolution of the vertebrate adaptive immune system, then is the requirement of the host to maintain co-evolved communities. And so rather than thinking of the immune system as a non-self-recognition system, I think that we might now begin to think of it as a management system. And I think, with all the data that are out there now, to me, that's a much more parsimonious explanation. So then, just to reiterate there, that the invertebrates seem to be restrictive, whereas the vertebrates are permissive. And the immune system, the selection on the immune system in the invertebrates may be for non-self-recognition, whereas the control of communities in the invertebrates. So this is that triumvirate I talked about. And so I just want to finish by saying that I think both bioinformatic approaches and functional studies provide evidence that animal-bacterial interactions involve ancient conserved characters. And so microbes have been around. The evolution of animals has occurred as a patina upon the microbial world. And so, in fact, it's not surprising at all that these are ancient conserved characters. And our new knowledge of the patterns and prevalence of these interactions demands, in my mind, that we question our very basic premises on the form and function of the immune system. So with that, I'll finish.